Welcome to Automata's podcast series, where we sit down with the movers and shakers of the tech and biotech industries to uncover the latest trends, insights, and innovations that are shaping our industries. My name is Lara, and today I have the pleasure of hosting a very special guest, Mr. Will Ferguson. Will is currently the VP of Pharmaceutical and Biotech Collaboration at Illumina. Will, great to have you with us. Could you start perhaps by telling us a little bit about yourself and the work that you're leading at Illumina?
Yes, happy to be here. Thanks so much, Lara. So my remit at Illumina really is to drive large-scale data generation from discovery cohorts, driving genetic evidence and genomics into in drug discovery. And we've found that having genetic evidence early on really does yield better results in the clinic and ultimately for patients. And so my remit is really from a corporate and business development perspective is to interact with the market leaders, both from the pharma and biotech side, but also the tools providers. So I oversee software partnerships as well, and then ultimately insight generation that will drive the next big breakthrough. So I'm focused on the partnership aspect of all of those things within Illumina and work cross-functionally across the organization with R&D, commercial, marketing, et cetera. Been in the genomic space for a number of years, going back to the NIH right after the genome was sequenced. And so it's been a lot of fun to be in the space.
That's great. You've really been with it for a very cool period of genomics. So that's really exciting to have come from where you were to where you are now. It's been exponential. That's very cool. You touched briefly there on partnerships. So impactful partnerships in pharma and generating meaningful data. I think this is something we're probably both quite passionate about. Can you walk us through some of the partnerships that you've been involved in and their impact on the integrity and the quality of that data?
Absolutely. Well, first I'll point to one that I wasn't directly, maybe indirectly involved with, and that's the UK Biobank initiative. And it started with the sequencing of 500,000 exomes, and the pharma industry partners really kicked that one off. And then shortly after, there was a public-private element to it where the whole genomes were also sequenced. So there's been some great comparators, and actually a recent paper that published comparing the whole genomes to the exomes, the pluses and minuses, et cetera, et cetera. But I would say the UK Biobank has been the 500,000 whole genomes and exomes linked to self-reported information, not the full medical record, but still nonetheless phenotypic data that is represented by a large population, in this case, within the UK, primarily Europeans. Nonetheless, 500,000 is a large cohort, and it's across disease areas. And so that initiative has really, I think, been the seed for lots of other similar projects within the United States, all of us, sequencing 1 million genomes. There's also now the follow-on to UK Biobank, or at least somewhat linked, but different, which is Our Future Health. And then Genomics England even predates UK Biobank, which is 100,000 genomes some years back, and Illumina has been directly involved with that as well. So these initiatives, there's more sites in Singapore, which is 100,000 Singaporeans that are getting a whole genome sequenced. We're directly involved with that. So essentially, the globe now has various projects around the world doing these types of efforts. And so, we're seeing this public-private kind of synergy, if you will, where there's some shared investment in resources, both financial as well as human capital and sort of the intellectual firepower needed to get value out of the data. But that is something that we see as only going to grow over time because the larger the sample sets get statistical power, and you can actually get to meaningful insights, associations within the data, et cetera, depending on the use case. But those are just some of the projects, some of which I've been directly involved in, others more indirect. But from a partnership perspective, it requires multiple companies, multiple types of organizations driving those. And it's really, I think, fantastic for the field and for science generally.
No, that's great. It's really interesting that you touched on sort of the different demographics there as well. And I think it's really wonderful to see that it's no longer just sort of one area focused, like you were saying, and this is really becoming a global demographic where we're able to pull data and understand across multiple different areas around the globe and the demographics that that are situated in those areas. So that's really interesting. Thanks.
Yes, yes, and other continents as well. I'll mention Africa, the Middle East, others. It's a global phenomenon, and it's happening all over the world, and it's very exciting. So yes.
Great, fantastic. You briefly mentioned AGD. So for those who may not be familiar with what that is the Alliance for Genomic Discovery. This is just a huge collaboration effort. What strategies have been successful in managing those efforts across such large organizations? If you could give us some insights on your personal experience there.
Absolutely. Early on, I've been Illumina almost 3 years now, and early on in my tenure, it was one of the first partnerships we were evaluating was ways in which we could help unlock and catalyze large scale data generation with partners, data partners that have potentially things like longitudinal health record information, and using that, again, taking a page out of the UK Biobank book and sort of that project, and hey, or can we drive other initiatives, maybe more patient in the case of UK Biobank, there are individuals in there with diseases of different indications. However, it's primarily healthy middle-aged Europeans. Again, phenomenal data set, amazing resource, but we're looking for other maybe complementary data sets that we could help catalyze and from a partnership perspective, maybe drive and sort of unlock. And so we looked at ways in which we could be more directly involved. So we sought out and built a strong partnership through senior executive connections and just a long-term relationship with Vanderbilt University Medical Center, the UMC, and their subsidiary Nashville Biosciences. And so we began to say, hey, could we do something together, the 1 + 1 = 3 or 5 scenario, and really maybe do something that we could bring to in a pre-competitive alliance consortia type way, similar to UK Biobank, but a little different in the ways that it's different, which I'll touch on in a second. And we said, yeah, let's do this. And so with some great interactions with those teams and just kind of a great chemistry that we had and built a strong relationship that led to the announcement that, we are forging this partnership. And then we followed with the first pharma to start and really kick off the Alliance for Genomic Discovery was Decode Amgen and sequencing the 35,000 primarily African descent individuals of African descent from that health system, and then subsequently brought on the other pharma partners throughout the year last year and made the subsequent announcements that you mentioned. But that was something that, that level of diversity from a whole genome sequencing perspective married to de-identified longitudinal health record data had not really been done up to this point. And so there was enormous value, scientific value, and opportunity to understand diversity in the genome in a bigger way. And so there was that interest, and that really helped drive at least, the kickoff or get things going. Having, the number of organizations involved, both Illumina, VMC, Nashville Biosciences, and then the 8 biopharma that are now part of AGD, it was required a fair amount of orchestration and coordination. I would say one of the biggest aspects of it when you're talking about investment in R&D, in the valuable R&D dollar, where 90% of drugs fail, it's actually 94%. And so we're looking at genetic evidence as a way to help increase the likelihood of success. You understand the underlying disease biology in a more in a deeper way, and you can create therapeutics that are more efficacious, more safe, et cetera, et cetera. And so, excuse me, I didn't mention this, but I will. It's often referenced, which is the seminal 2015 Nelson et al. Nature Genetics paper, where he showed while he was Matt Nelson was at GSK leading genetics, he had actually shown that therapies with genetic evidence early had a 2x likelihood of getting through the clinic and getting to FDA approval. And subsequently, we've seen in the last 10 years over 63%, and this was published in the fall in Nature last year, that 63% of all FDA-approved drugs in the last decade actually had genetic evidence. And so we're seeing this shift in the industry to more and more scientific leaders in biopharma now making these investments early in R&D, getting to genetics, leveraging genetics and genetic evidence in different ways to drive impactful discovery and more efficacious therapies down the road. And so that was like one of the key anchors that we leaned on for pitching this idea of the 250,000 whole genomes from the VMC Health System from Nash Bio, and really helped us differentiate that cohort versus the UK Biobank in the sense that it's more diverse, neonates to geriatrics, so the age, it's diverse on multiple dimensions on ancestry. 15% African American ancestry in there, and the 70% range for Europeans, so it's more diverse. And you also have de-identified EMRs, so the full EMR, so it's longitudinal, on average 11 years of longitudinal health record information per individual. And then you also have, these are from patients, right? This isn't just healthy individuals. So you have disease diversity, a large cardiometabolic swath of indications represented there, as well as neuro and other indications. And again, these were DNAs from discarded blood samples from routine lab practice that they bank as part of their system. So it made a lot of sense. And so that's where some of the key, I guess, parameters are that we considered when going and talking to R&D leadership across pharma. We knew that going in, that for them to make the level of investment that was being contemplated, there needed to be a clear return on that investment, not just for the greater good, which it definitely is that, but more importantly, to really help them find better, more efficacious targets, and also prioritize the ones that they have and leverage data to do that.
Oh, wow, that's very insightful. Now, talking about all of these different I don't want to call them data sets, but groups of data that everyone's collected between the different biobanks and all of these different areas. Have you, as Illumina, or as a group of people working in genetics with the same mission, have you seen a sort of standardized data format or set that everyone's running towards, working with, in agreement with? We've seen this across so many industries where formats and data standards have been a huge debate and often a huge blocker in terms of sharing information with one another. Really interested to understand how you have overcome that, especially now that it's very much global practice and it's not just within one country or within one single area.
Yes, it's a great question and a really complicated topic generally. What I will say, even before you get to the data itself, one of the most important, I guess, points to consider is how the individual patient samples were consented. So consent in the proper IRB protocol within the institution. And again, this could be an academic medical center that's linked to a health system or a hospital. It can be just a research lab that works with that has docs that do, it can have all these different flavors, health systems, AMC, academic medical centers, et cetera. Regardless, they have to have, in order to use those precious samples for research purposes, they have to have the proper consent in place. And that consent language and again, within each country, there's some variation, but overall, it needs to be protected. And in US, you have HIPAA, you have GDPR now in Europe. And so the consent language for the samples themselves have to be, that protocol has to be airtight and really done in the proper way. Once you're past that, then you get into, okay, here are the sample types. And again, these biospecimens, you may have whole blood, You may have just DNA that was isolated downstream. You may have tissue if you're talking about oncology indications, et cetera, not just oncology, but that primarily that's one of them. So you have these different types of samples. And so the different molecular data you're going to generate from those samples will dictate in kind of what research is being contemplated, will drive the standards and kind of what ends up being implemented. But I will say that from a germline genetics point of view, that 30x coverage, whole genome has really, and I think the standard has kind of been set there for research. At least I would make that argument. Some might challenge me on that. But if you're looking at large discovery cohorts, getting a 30x whole genome, and you do mapping and alignment, we use the Dragon pipeline for secondary analysis. It's industry leading on the precision FDA. It is something that we believe and support. You end up with a VCF, which is a variant call format file, and so that is kind of a standard way to interrogate this type of data, and then you look to the phenotypic data that you're going to marry to that. That could be structured clinical data, ideally from the EMR directly de-identified. And so that has to be in a, depending on the, the consent language that you have, of course, if you have it consented properly, then you can provide that for research purposes. It's fully de-identified. And so that can be used for whatever new breakthrough discoveries that will come. If you're in a European setting, you need to have GDPR compliance in place, and the technology you're using, platforms, cloud, on-prem, regardless, you have to sort of follow these rules and make sure that you're operating, as you need to. But essentially, it's still early in the discovery cohort realm. I think I would say that type of data at that depth of coverage, those files are fairly standardized now. I would make that argument. But when you look at other areas, you look at proteomics, you have different platforms that are still out there. Some have been used for the larger population projects. For example, the UK Biobank, they did a subset of 50,000 proteomes of the PPP project, the Pharma Proteomics project that was published in Nature, some of the results from that, which is really exciting, linking that to the variants PQTL, which is protein quantitative trait loci. That's an application of a lot of interest within pharma, but that's, again, a new modality. You need to have serum to do that, right? So it really is, I think, based on the resources and the specimens within the institutions, and then what the research goals and the scientific questions that are going to be asked, and sort of linking those together. And then the output from that, of course, has to follow the frameworks within those protocols, and make sure everything is, kind of done in a proper way, like I said, around consent language and the like frameworks.
Oh, hearing all of that, it seems like you're a lot further along than a lot of different other industries. So it's great to hear that there is an overall consortium of rules and regulations that sort of umbrella everything when it comes to those patients' rights, data privacy, and equitable access to research. So good on you.
Yeah.
Okay.
It's evolving. I would say it's evolving, and there's an intent, and there's some frame. It's still early, but it's not as there are some key pillars that are in place. I would totally agree with that point.
Great. That's good to know. Again, way further along than so many other sort of areas of research where no one can seem to agree on formats and rules and regulations. So that's good to know. Moving on, the impact of genomic data, sort of the deluge on drug discovery. What would you sort of say in terms of what that deluge of genomic data is that's being generated at population scale now? So this is really, really huge. What is the impact that is anticipated on drug discovery in the more long term versus what's really happening now?
Yes, I think I think a lot of people will tell you that, and if you look at some of the KOLs in our space, I mentioned The Nelson et al. Paper a few minutes ago, like Matt Nelson, who's now at Deerfield leading genetics and genomics, or Mark Daly, these types of, sort of leaders in, he's leading up the FinGen initiatives at the Broad Statistical Geneticist that's well known and respected in the whole industry. A lot of these thought leaders believe there's a lot of, that there's a lot left, a lot of green fields as it pertains to genetic targets in complex disease in areas where we haven't had data at this scale. And so getting to the levels of statistical power by disease area from patients doing these large GWAS, FEWAS analyses at the scale that we're doing now, it's just something that hasn't been done. And so there is a feeling that we will find some really impactful breakthroughs. If you look at recent, one of the staples of, or often referenced success stories of the genetics is PCSK9, which is around cholesterol, and that was a target that was found and subsequently has therapies that have been designed against it. It's been very successful, and the mechanism was clearly identified and leveraged using genetic evidence, and that's kind of the gold standard. And so looking for other targets like this, again, where regardless of the therapeutic modality, having data at the scale that I've referenced, being able to get to the signal and understand those associations with deep clinical longitudinal health record information and a whole genome sequence, not a genotype or other modality, but something as deep and informative as whole genome sequencing, that really we do expect breakthrough discoveries in these indications that have been really challenging in neuroscience, in cardiometabolic disease, across the board. We really anticipate and expect breakthrough discoveries coming on the heels of this deluge of data that's been generated. I do think it's worth mentioning, however, that just because you generate the data, that doesn't mean the themselves. You have to have the infrastructure and the intellectual firepower to get value out of that data. And that's something that is really, really important for not only the biopharma researchers, but also those on the academic and nonprofit side as well. As it's our training and how we're looking at these data is making sure that we have, the statistical geneticists, the bioinformatics folks, the researchers out there to really get value out of this data, and it will help ultimately patients and potentially get to cures, in the nearer term for a lot of these really, very debilitating diseases that the globe suffers from. And so there is anticipated breakthroughs on the horizon, and a lot of thought leaders across companies, nonprofit, profit would I think agree with me on that point. And it's very exciting. So we're excited about not only the breakthroughs, but all of the interesting, how we're going to understand the fundamentals of disease biology and the mechanisms of action because of all the different layers of molecular data that we can generate for patients at the scale that we can generate now. At the cost, of course, has come down significantly, so we can get into these large scale projects. And so, no, I think really the sky's the limit on the breakthroughs that we anticipate. It's just a matter, it's a matter of if, but a matter of when.
Yeah.
But no, it's very exciting for sure.
Yeah. Super exciting. So in closing question, in hypothetically 5 years from now, Will and Lara are sitting back having a conversation on the same sort of topics, what would be, in your eyes, the optimal place for us to be 5 years down the line, continuously or continuing the good work that we're doing now within this space?
That's a great question. Aspirationally, if we had whole genome sequencing in the clinic globally for every human, I think that would be amazing. Again, that's very altruistic.
Yeah.
But that would be an amazing feat if we could get to that, where the health systems and the governments around the world supported that because of how informative and useful it is over the course of a person's lifetime. That would be, I think, very impactful if we eventually got to that point. And also, of course, discoveries in some of these really challenging diseases where we're going to start to see numbers in disease areas like whether it's Alzheimer's, ALS, a lot of the neuro indications, which are quite challenging. And we know they're polygenic and they're complex, and we don't know that much. But in 5 years, if we have made some real breakthroughs scientifically in those areas, would be amazing. If we actually did have, say the PCSK9 and Alzheimer's or disease XYZ that's quite challenging, where we really don't have any therapies. But having those in 5 years, that would be, or at least on the way to patients. I mean, the process takes time, but it would be amazing if we did have some breakthrough discoveries that are really, ultimately helping patients. That would be awesome.
Yeah, I share the same hope. So I will pencil you in for 2029, and hopefully we're sitting in a good space. Thank you so much, Will. This has been great to have this conversation with you. And if there's anyone that is interested in joining the Alliance, wanting a bit more information, where can they go after listening to this to find more information?
Yeah, I mean, they can contact, Illumina or Nashville Biosciences directly. We do have, there is a site that the Nash Bio team put up, and it's just nashbio.com, nashvillebiosciences.com/AGD. And so that's one immediate place to go for quick. There's videos and just an explanation of the Alliance and the structure. And then, of course, they could feel free to reach out to me on LinkedIn or any other folks at Illumina directly. But yeah.
Fantastic. I think we'll link it in the notes under each of these recordings as well. Spread the word and keep up the good work.
Thank you so much, Lara. Enjoyed it. Cheers.
Thanks. Cheers.